Hi, my name is K Esoh. I'm a PhD candidate in human genetics at the University of Cape Town in South Africa. I'm pleased to share our work entitled Genome-wide Association Study Identifies Novel Candidate Malaria Resistance Genes in Cameroon. Let me start with a quote from a 1971 review by the American biological anthropologist Frank B. Livingstone that malaria has probably killed more human beings than any single disease. Malaria is caused by Plasmodium parasites. It is transmitted to humans by female Anopheles mosquitoes. The deadliest form is most common in sub-Saharan Africa, where children below the age of 5 are the most vulnerable. Given the high mortality rate today, we can better appreciate Livingstone's words by considering the world before the discovery of the most effective anti-malaria drugs. Certainly, malaria has been a major evolutionary force on the human genome for the last 5,000 years. This pressure has resulted in many susceptibility markers, which are now known to account for up to 25% in the difference in response to severe malaria among individuals, of which the sickle cell mutation of the HBB gene contributes the largest effect. Much of these genetic variants are shared across sub-Saharan Africa, yet some are region-specific, for example the HBC in West Africa and the Dantu in East Africa. Cameroon, located west-centrally, exhibits some unique patterns, such as the weakest sickle cell mutation protective effect and absence of other genetic variants or associations. But data suggest malaria and the sickle cell mutation might have originated around Cameroon, meaning the malaria selective pressure should have acted longer on Cameroonian genomes. But we have also characterized fine-scale genetic substructure within the country that might have a significant impact on genome-wide association studies. This therefore raises the question whether Cameroonians harbor other malaria protective loci whose detection might have been hampered by fine-scale population structure in the country. To investigate this, we used case control data of Cameroonian participants that contributed to malaria gen projects. Quality control was performed, followed by imputation by 4 strategies using 3 reference panels, and association analysis using 2 strategies, first on the pooled data and then by ethnic group. Lastly, haplotype analysis was performed. We observed a higher haplotype conservation in genomes carrying the sickle cell mutation, consistent with increased evolutionary pressure. Subtle differences observed among the ethnic groups was consistent with fine-scale genetic structure, suggesting different patterns of association might be expected. Indeed, there was disparate patterns of association among the ethnic groups. A signal on chromosome 10 in the pooled data was observed only in the African Genome Resource Panel from the Senga Imputation Service. Another signal on chromosome 6 was observed only in the semi-Bantu. I will focus on the chromosome 10 locus, whose biological role and expression pattern suggests it might be an attractive target for adaptive evolution, especially in malaria endemic regions. The lead SNPs are highly specific to malaria endemic populations. Tag variants of the lead SNPs are strong EQTLs in the liver, where Plasmodium parasites spend much of their time in humans. This raises the question whether the lead SNPs are under positive selection in malaria endemic populations. However, alignment with other primate species suggests a purge of the SNPs in most populations. Selection and haplotype analysis suggest that the SNPs are most likely to be under a recent balancing selection in malaria endemic populations. Chromatin-stated data suggests the SNPs are located within a region that is active in human embryonic stem cells and polycomb repressed in liver cells. Transcription factor binding site data revealed some of the SNPs disrupt the binding motifs of certain transcriptional activators. In conclusion, given the association of the locus with the progression of certain cancers, we propose the variants herein identified might have been purged from most populations to reduce the gene expression. An opposing force, such as protection against malaria, therefore seems like a reasonable explanation for the persistence of the variants in malaria endemic populations. I would like to give special thanks to the ICB and the GLBIO for the travel fellowship that enabled my presentation to be possible. Thank you for your keen attention.